Good evening, everyone. I hope that you are all well. Okay, good afternoon or good evening. It's a very hot Saturday, so I first of all I would like to apologize for not having class for the past 2 weeks. Okay, because I was quite busy. So today I'm continuing chapter 4, which is 4.5 nucleic acids. Okay, so actually I know that the syllabus. I think most of you are quite far ahead. You probably would have been learning chapter 6 or 7 or even 8. Okay, but doesn't matter. This is for a revision for yourself. So if you have any questions, I'll be most happy to answer your questions. Okay, so let me share screen. Let me see who is over here. All right. Okay, Happy Lee. All right. Okay, good afternoon. All right, so I'm going to share screen now. I hope you can see this. Okay, today we're going to learn about the molecule or the structure of nucleic acids. Okay. Usually nucleic acids will be represented by this very diagram that we always see, right? The very famous diagram. Actually, what you see here is the DNA molecule. Okay, DNA molecule is one example of nucleic acid. All right. Okay, so like I said, one example because that's not the only one. Okay, now let's start. Yeah. All right. So let me see where's my. Oh, all right. Okay, so this is the first screen. All right. Now the second slide here. Now you know that this molecule is what I've shown just now earlier. So this is actually the DNA molecule. Okay, it is a molecule which carries genetic information. Genetic information, that means it has information to produce proteins. Okay, and the proteins, as you know, is the basic building block of life. Okay, a lot of enzymes, a lot of these substances which are protein based. All right, you need the protein to produce all this stuff. So the formula, all right, or what we call what they call the code, right, to make the protein is actually found in the DNA. So it carries a unit of inheritance, right? So it passes down information from a cell to the other cell, we call it the offspring. Okay, so let's look at what is nucleic acid, right? Now, nucleic acid is formed from, actually you will see this polymer chain. So what you see here is this the polymer chain, all right? Here, this is the polymer chain. You see two chains here, right? They are intertwined. So it can be one or it can be two. So if you see that there are two polymer chains, this is a DNA. Okay, the other type of nucleic acid is only one polymer chain. The one polymer chain, the example of the nucleic acid is called RNA. So that means there are two types of nucleic acid. One is RNA and one is DNA. If it's only one polymer chain, it's called RNA. If it's two polymer chains, we call it DNA. Okay, so what you see now here, you see two polymer chains intertwined, right? And you call this DNA, right? Now, first of all, what is the basic unit? How do you make this molecule, all right? You can see that it's a continuous structure, right? The basic unit is actually repeated. Okay, the basic unit to form the DNA molecule or RNA molecule is the nucleotide. Okay, and nucleotide looks like this. There are three components to the nucleotide. Okay, you will see that you have the phosphate group. Pentose sugar is a type of sugar which is 5 carbon. There are 5 carbons there, right? C5, right? And also a nitrogenous base. Okay, you do not need to know the structure of these three components. All you need to know is there are 3 parts of this in order to make a nucleotide. And in order to make your DNA, this nucleotide is repeated. Right? It's repeated, so it becomes very long. When it becomes very long, it's called polynucleotide, and that forms your basic backbone. That forms your one of the chains. Okay, what are the elements that are in the nucleotide? Okay, what is the basic elements? In order to form DNA, what are the substances or we call elements that you need? Okay, so you will need these elements: carbon, hydrogen, oxygen, nitrogen, and phosphorus. These are the elements that are needed to form your nucleotide. For example, you see the P phosphorus because you have a phosphate group here, right? You can see the P here. Here, right? There's a P here. Then of course here we have C, right? Here, wait, down here. But over here you have C here, right? C, and another C here, right? There's another C over there. Then you will have oxygen also in the molecule there, oxygen and hydrogen, and also nitrogen. Okay, nitrogenous base means there's nitrogen there. All right, okay, so remember CHONP. All right, okay. So that is the basic unit. You need to recognize the basic unit of DNA or RNA is the nucleotide, which will be repeated, okay, in order to form the long chain. We call the polynucleotide chain. Okay, if you have any questions, you can just type in. Okay, type it over here and I'll try to answer the question. Okay, let's concentrate on the nucleotide, yeah? All right. So nucleotide. Now, the basic, as I said, the basic unit. Monomer means it's the basic unit that is going to be repeated. That's going to be repeated. So you call the basic unit or the building block. The building block of the DNA or RNA is the nucleotide. Okay. Now, there are three parts. Okay, the first one is the phosphate group, where you have the PO4, right? Then there is this sugar. Okay, it is bound together with a pentose sugar, which has got 5 carbon. So it's called a 5-carbon sugar. Okay, unlike your glucose is a 6-carbon sugar. So pentose is a 5-carbon sugar. Okay, and then you have this nitrogenous base. So nitrogenous base is a general name. Not only one type of nitrogenous base, there are actually 5 different types of nitrogenous bases. Okay, pentose sugar, there are 2 types. Phosphate group, there's only 1 type. Okay, there's only one unit, 1 type of this. There's no variation for your phosphate group. But pentose sugar, there's variation. There are 2 types of pentose sugar. Okay, and the nucleic acid that is made, what kind of nucleic acid is made, it depends on the type of pentose sugar that is there. Afterwards, I'll explain. For nitrogenous base, there are 5 options. There are 5 different nitrogenous bases that can be attached to this nucleotide. Okay, so you need to recognize there are 3 components for your nucleotide. Always you will draw the phosphate group as a circle, the pentose sugar as a pentagon, 5-sided, and a nitrogenous base, you can just draw a rectangle, or you can draw a rectangle with a certain shape at the other side. Okay, it doesn't matter. Right, so let's go through. Just like I mentioned, there are 2 types of nucleic acid. Okay, one is DNA, one is RNA. So what type of nucleic acid is formed depends on actually what type of sugar there is. So pentose sugar, just like I mentioned, there are 2 types of pentose sugar. Okay, so let's see what are the 2 types of pentose sugar. All right, okay, the first one is called, this is the pentose sugar, right? So there are 2 types of pentose sugar. One is called a ribose sugar. Do not worry about this, how to draw this, the molecule, the structure, and so on. You are not needed in Form 5. Okay, so you will see the other type is a deoxyribose. So the difference is only at one place. You will see the difference is only here. This one has got OH. Okay, that means there is an oxygen there. But here there's only H, there's no O. So they call it deoxy. Deoxy means it's lacking of one oxygen there. Okay, deoxy, that means it's already removed 1 oxygen. So the difference is only whether there's an OH here or the other one is just the H. Okay, how to draw this? Where is your C? Where is your O? Where is the H? It's not needed. All right, okay, so you just need to know it's a pentagon shape. And the 2 types of sugar is a ribose or a deoxyribose. Okay, so now, what type of amino acid is made? What type of nucleic acid is made depends on what sugar is present in the nucleotide. Okay, so I let you know. So there are 2 types of nucleic acid, all right, depending on the type of ribose sugar, pentose sugar. Okay, so if it's RNA, we call it, RNA stands for ribonucleic acid. So you see the word ribo, that means the sugar here, right? The other one is called deoxyribonucleic acid. So DNA, RNA and DNA. So the difference is actually the only difference is the type of sugar. For RNA, the sugar that is for the pentose sugar is ribose sugar. This one. Okay, no need to remember the OH, but it is a ribose sugar. But for the deoxyribonucleic acid, the sugar that is used to make that pentose sugar, right, is deoxyribose. It's this one. Okay, and there's also one difference. Another difference is the nitrogenous base. Okay, for ribonucleic acid, it can be A over here, it can be C. Okay, what is A, C, G, and U? These are different types of nitrogenous bases. I will show you in the next slide. Adenine, cytosine, guanine, and uracil for ribonucleic acid. And for the DNA, is either the A and the C, the same thing, G, but this one is yeah, thiamine. Yes, either A, C, T, or G. So the nitrogenous base is also different. Okay, for deoxyribonucleic acid, there is no uracil. There's A, C, T, or G. That means it can only have one. Okay, that means there are different options. Okay, phosphate group is the same. Deoxyribose sugar, all right, the oxyribose sugar is also one type, but the nitrogenous bases, it can be either A or C or G or T for your DNA. For your RNA, it is ribose sugar. Phosphate is the same. Nitrogenous base can be A or C or G or U. Okay, so made up of these components. Okay, so I'm going to show you now the nitrogenous bases. All right, so nitrogenous bases, there are actually 5 types. Okay, the first one is called adenine. Again, you do not need to memorize how to draw it. Okay, what is this N, where is the H and all that, no need, right? This is adenine, represented with the letter A. Whenever you see A, you think of the adenine. Then G is guanine, okay? Then the other one is thiamine. T stands for thiamine, okay? C stands for cytosine, all right? Now for DNA, you have only these 4 options. DNA, you have A, C, or G, or T. A, G, T, or C. But for RNA, okay, you have either A or G or C, or the other one is U. Okay, don't have thiamine for RNA. All right, you can see there are options. The nitrogenous base got different options, different types of nitrogenous bases. Pentose sugar either is ribose or deoxyribose. Then for phosphate, it's the same. Okay, there's only one. There's no variation for the phosphate. Okay, so far okay, right? So no questions here, then I'll carry on. All right, so this is a revision. I'm sure most of you have studied this. Okay, I just want to finish, complete my catalog of lessons. Okay, I do not want to leave it hanging at 4.4 and not finish. So I'm finishing this. Later on, I'll go faster and maybe I'll complete chapter 6 and chapter 7. All right, okay. Now let's compare the DNA and RNA. Right, comparing means you must talk about the similarities and differences. Okay, so both DNA and RNA are made up of this nucleotide. Both will have this basic unit of nucleotide. Okay, and you can see the difference here. Look at the strand. Okay, for DNA is 2 chains, double. DNA is double, right? Double chain, right? There are 2 polynucleotides. But for RNA, it's single. There's only 1 chain. Okay, they don't have another chain that complements it. All right. So this is a difference. The similarity, just like I mentioned, both are made up of nucleotides. Okay, but the sugar is different now. All right. Now, so this is called the backbone. The phosphate group and the pentose sugar is actually the backbone. So if I simplify it, this will be the backbone. Okay, this is the phosphate. Let me draw this. This is a phosphate. P is a phosphate. This is your ribose pentose sugar. Okay, this is your sugar S lah. I'll put S. Okay, this is your S. This is your sugar. So repeated P and S, phosphate sugar, phosphate sugar, phosphate sugar. It forms this part. This is your backbone. You see the ribbon here? That is your P and your S and the P and the S continuously. And same for here. This is also P, then you have the S, then the P and the S continuously all the way. Okay, continuously. What is different is every one of the nucleotides may have A, all right, and this side will be T, okay, and this may be C and this may be G. So you see the nitrogenous bases may be different, but your phosphate and sugar right? That forms the backbone. This is repeated. It's the same for the backbone. All right, okay. So let's look at this is the phosphate sugar backbone. Okay, the ribbon that you see that is a P and an S, P and S, P and S. All right. So and then the middle part here, you'll see this like ladders. All right, the ladders inside here. This shows your nitrogenous bases, which are always paired for DNA. You will see one side is one color, the other side the other color, because they show them complementary base pair. Adenine will always match with your thymine. And then cytosine will always pair up with your guanine. This is a pair matching. So normally when we draw, we will do the complementary shape. If this one curves inwards, that one, the other side must be curved upwards to show its complementary pair. Okay, for the RNA, there's no complementary pair. It can be A or C or G, but there is a U there. Okay, for RNA, there's the U there. A or C or G or U, no T. For DNA, A or C or T or G. Okay, so here, DNA, there is adenine. RNA also have adenine. Guanine for DNA, this is similarity lah. RNA also will have guanine. Cytosine for RNA, this one, DNA. RNA also will have cytosine. Okay, but this is a difference. DNA will have thymine, but RNA will have uracil. Okay, so you can say both are made up of nucleotide basic units. Both have adenine, guanine, cytosine as nitrogenous bases. Okay, and differences. DNA has thymine, but RNA has uracil for nitrogenous base. Okay, not? Okay, so this is basically you can see here the differences and the similarities between DNA and RNA. Okay, right, I go on, yeah. So next page, you see this is the structure of DNA molecule. You see the backbone, right? The backbone, this is your nucleotide basic unit, which is the phosphate group, deoxyribose sugar, and your nitrogenous bases. So it's always T and A or A and T. They always, we call it complementary pair. And there is hydrogen bond that bonds your two complementary nitrogenous bases together. Okay, it is like facing inwards. Okay, so the outside here will be the two polynucleotides, which is the backbone, the sugar phosphate chain. This is for DNA lah, right? And then the inside further facing each other will be a nitrogenous bases. It's always A and T or C and G for your DNA. Okay, clear? So, right, this is I think from your textbook. Okay, okay. I'm not sure. I think. Now next one. So let's look at again. Let's look at the DNA. How to describe the molecule of DNA? So you will say it consists of two polynucleotide chains. Okay, you can see two ribbons there. Okay, don't use the word ribbon. Ribbon is for you to visualize. It looks like ribbons, right? You don't say ribbons. Okay, what is the term to say this? You use the word double helix. Okay, they are intertwined. Intertwined is like berpintau, pintau, you know, DM. It's like you wrap it around each other, but in opposite direction, you know. Afterwards, I'll show you what it means by opposite direction. One is coming down, the other one is going up. So the direction is actually opposite. Okay, so do not draw it like mirror image. It's not mirror image. A lot of students make the mistake of drawing one side and then the other complementary side, they just flip it over and then they draw the same thing, which is just the other side, which is wrong actually. Okay, so this is double helix structure. Okay, this is how you explain, name it, you name it. You don't say ribbon now. All right, then this is how it looks like. So this is your sugar phosphate chain. Okay, you see that purposely put terbalik. Okay, it is upside down for this side. Okay, this is your D stands for the deoxyribose sugar, the pentose sugar lah. P stands for the phosphate. And then on the other side, you find the direction or the orientation is different. And it's like facing up. The other is facing down. All right. And this is the backbone. And the complementary nitrogenous bases, because we're talking about DNA, is either A, C, T, or G. They are bound by hydrogen bonds. For C and G, there are 3 hydrogen bonds. For A and T, there are 2. This is not important for the form 5. Okay, but as long as you know there are hydrogen bonds that binds your complementary nitrogenous bases. A always with T, C always with G. Okay, clear? Okay, next. So now look at the sugar phosphate backbone. It's like the ribbon there, right? And then in between you'll find your G and your C, okay, and your T and your A, or A and T. So we consider this how you want to describe the shape. Always use the same, the correct term for it. It's called a double helix structure, made up of two polynucleotides intertwined, forming a double helix structure. Okay, so they're intertwined in opposite directions. So we call it anti-parallel. Okay, that means it's not exactly if you flip over to the other side. For example, okay, this is what's a passive question now. So if I give you this side, okay, and the question asks you to draw the complementary phosphate, the chain, the polynucleotide chain for the other side. Okay, so most students will do this. All right, they will just have a mirror image of the phosphate, the what you call the pentose sugar with the phosphate group. You just mirror image over to the other side. All right, and then of course they remember this one. The nitrogenous base, you must use the complementary. If this is an arrow facing this way, then this one must have this one is jutting out, then the other one has to be like that. You have to draw like that, okay? And then if then is curved like this, then this one have to curve the other way to show it is complementary base pairs. Okay, and then this one like this, all right, and this one is like this. This is correct. But unfortunately, there's one very serious mistake with this. It is the direction of your sugar phosphate backbone. The one on the left is like this, right? It's facing up. The other one is not facing up. Okay, so this is what the students will always make. Okay, I'll rub this away. It is like this actually. You want to draw it, you have to flip it over. So I always tell my students, in order, and I'll do it again, okay, like this, okay, you have to flip your paper. If you want, you're used to drawing like this with the pentagon facing upwards, you find it difficult to draw the other side. So what you do is, I always tell my students, when you want to draw, you just flip your paper over, you draw exactly the same thing, okay, and then when you flip it back, it's the opposite already. Okay, you terbalikkan your paper, and you draw like normal, and then when you flip it back, it is terbalik already. Easier to draw that way. Okay, so this is the correct one. And where it joins must be correct. It joins like this. Okay, join over here. Not over here. A lot of students are going to join like that. Wrong. Not over here. Okay, not the first, not the carbon number, we call it 5. Okay, it is 3 and 5. Carbon number 3 and carbon number 5. Okay, clear? All right. So I go on. We call it anti-parallel. Now, so you see like this. This one is, you see this one facing up here, right? All right. The pentagon is pointing up here, and then the other pentagon is pointing down. Okay. And then you remember this one must be correct, this one. Okay. A and T, C and G must be like that. Okay. That is your structure of DNA. Right. So you see this animation, GIF of, you always see this thing spinning and spinning and spinning. This shows the DNA molecule. Okay, with all the little things showing your atoms. The either is carbon, hydrogen, oxygen, nitrogen, or phosphorus. Okay, all right, clear? I hope I've explained this well. All right, so you won't have any ambiguity about DNA. Okay, let's go on to the next one, which is the RNA. RNA is ribonucleic acid. Okay, so it's a single polynucleotide chain. No, you don't have the complementary base pair. There is nitrogenous base, but it doesn't have a pairing. Okay, it will be shorter than DNA. So usually, you see the nuclear bases, or we call the nitrogenous bases, it don't have pairing. It's either the A or the C or the G or the U. It's not T. You have uracil over here, and the backbone is the same. You have the sugar phosphate backbone, same like for the DNA. Okay, so complementary nitrogenous bases are A, C, G, or U only. The U is to replace the thymine. So no thymine, all right? Thymine is replaced by uracil. Okay, so that's a uracil. Next, now, so this is what the RNA looks like, and there are actually 3 types of RNA. Okay, this polynucleotide not always is in linear form. Okay, sometimes this one in linear form is called mRNA. It's a messenger. M stands for messenger RNA. This is used to copy out the code from the DNA in the production of protein. When you want to make protein, your cell wants to produce protein, you have to copy out the code from the DNA. So your mRNA copies the code, okay, from the codon. We call it the codon, from the code from the DNA, and bring it out to the ribosome, because ribosome is the place where they produce the protein. So the other type of RNA is the ribosomal RNA. This is the the RNA that forms. Okay, this is where the production of protein, the synthesis of protein occurs, is at the ribosome. Okay, so there's RNA, rRNA actually, and then the other type is called the tRNA, so called transfer RNA. It transfers the amino acid. Okay, when you want to make the protein, it brings in the amino acid and then it joins up together, or the amino acid joins up together. I'll show you afterwards. Okay, all these three are involved in protein synthesis. So remember, your RNA is important. It will be utilized, it will be used to make all types of protein in your body. Protein including your antibodies, your enzymes, okay, or the amino acids and so on that you need to maintain, right, your health, and also to carry out certain processes in your cells. Okay, so there are three types of RNA. So the importance of nucleic acid, okay. Just now I've already mentioned earlier the DNA. It is the unit to carry your genetic, what you call the unit of inheritance, okay, your characteristics. It is a carrier of hereditary, or we call genetic information, okay, because this information that is found in the DNA determines what kind of characteristics you have. Okay, some people are born tall because you have the tall gene. Your father is tall, your mother is tall, let's say. Or you're born with very fair skin. Okay, in your family history, everybody is very fair because you have the fair genes. Or maybe you're dark, right? Your family history, everybody is dark because you have the lot of melanin production in your skin, or in your skin, right? You have a lot of these pigments, production of protein called pigments. So it makes your skin dark. So all of this is carried in the DNA of your body, of your cells. Okay, so it determines whether you are curly hair or whether you are straight hair, whether you're dark skin, light skin, all right? The type of blood you have, whether you are A or B, AB or O. All this is encoded inside your DNA. Okay, and that characteristic is going to be passed down to the next generation. Okay, so the trait of an organism, whether you have fair skin or dark skin, it depends on the type of protein that you produce. So if you are very dark skin, you probably have a lot of melanin in your body, okay, on your skin. Okay, you have a lot of melanin. Actually, melanin is also good. It is actually going to protect your skin from sunburn. Okay, so people who are dark, they are not so easy for the person to get sunburn compared to people who are very fair. Very fair, they can't stay out in the sun for a long time because they don't have the melanin, don't have enough melanin to protect the skin, the UV light and so on. Okay, so whether you have dark skin or fair skin depends on the type of protein that you produce. Okay, and the DNA will contain the code. Okay, the code is carried by the nitrogenous bases. Okay, and nitrogenous bases determine what type of protein you're going to make. Okay, I'll show you an example. Look at this. Now, this is your mRNA. Just like I said, your mRNA is going to copy the information from the DNA. So it copies out the information. So your mRNA will have this G and the C and the U and the A, right? The guanine, the cytosine, uracil, adenine, and then repeated lah, right? So this is copied from the DNA. Now it comes out, okay, and goes. You have learned it in. You haven't learned it yet, chapter 5. It's going to go into the ribosome. It's going to go. This mRNA will go to the ribosome, and it's going to produce the protein. Okay, what kind of protein are you going to make? For example, the first three here, GCU, is actually a code. Okay, we call it codon. The codon is a code. It's like a recipe to make a type of amino acid. So it's fixed already. GCU is going to make alanine. Alanine is one of the 20 types of amino acid. You have learned earlier in protein, your class on protein. There are 20 types of amino acids. So GCU is going to make alanine amino acid. Don't have to memorize GCU is alanine. No need to memorize this. Nobody in the world is going to remember GCU is alanine. Nobody have the time to memorize all this. Okay, they are not going to ask you. For example, ACG, adenine, cytosine, and guanine. These three, it forms a codon. This is a codon to make the next amino acid, which is called threonine. Okay, so when you have all of this produced, right, the third one, GAG, is going to make your glutamine. CUU is going to make leucine. CGG is arginine. Okay, AGC is serine, and so on. So now you have all these amino acids. What happens is we're going to join it all together. When you join your amino acids, you're going to make a protein molecule. Then you are making protein. This happens at the ribosome. Okay, so you can see the process of your nucleic acid producing different types of protein based on the code that is copied out from the DNA. Okay, so you have different amino acids, different code will make different amino acid. When you join together, you're going to make a molecule of protein. Okay, you remember, amino acids are the basic unit of making protein. Okay, so what kind of protein you're going to make depends on the type of codon, what is the sequence of your genetic code in your DNA. Okay, right. So here I'll show you what it means. Your DNA carries the recipe, the formula, right? All your A or C or T or G. Let's say this is the A, this is the C, this is the T, this is the G, this is repeated A, right? So this is your DNA. Then your mRNA will copy out. So when A, you copy out the U, it become the U here. Then the C, you get the G here, right? Don't worry about the order, just simply example only. So your mRNA now goes to the ribosome. This happens at the ribosome. Okay, just I want to give you an entire picture. You are not don't need to write all this in detail, but it will help you a lot if you understand this. You go to Form 6, you will know what it's all about. The whole story you will learn in Form 6. Okay, so this codon is going to code, right? It's going to make this methionine. So it's going to be translated. Okay, your AUG will be translated to form your methionine amino acid. Then your CGA is going to form your arginine amino acid. Then your UAC is a codon to make tyrosine. Okay, then after you join it all together, you will get a polypeptide. So there is your protein. Okay, so your RNA is important in synthesizing protein. Okay, you don't need to see the whole picture, but I want you to understand it. Okay, so here the three codes here is called the codon. Okay, you need the three codes here to make a amino acid. Okay, this is like a recipe right, to make the amino acid. So AUG codon on mRNA will code for methionine. The AUG will be able to make is a code to make your methionine. The base sequence on DNA is transcribed, is copied, right? It's copied onto your mRNA. Transcribed is copied, and then it's translated into this amino acid. Methionine, arginine, tyrosine, how it's going to make your protein. Okay, so this is what you see in your textbook. Okay, this part here. Because if I don't explain this, I quite unlikely you will understand. You will not understand what it's talking about. Okay, so I'm just trying to make it clear to you what you are learning, what you're supposed to learn. Okay, next. I think that's going to be the last part already. So the genetic code on the mRNA is translated into protein. Okay, so that's how you get your protein. It's all based on what kind of codon is in the DNA. You have to copy out from the mRNA. You'll see that again in chapter 5. Okay, so here now, lastly, how do you form chromosomes? Chromosome is the genetic unit, the inheritance. Okay, this structure inside the nucleus that is going to carry this unit of inheritance. Okay, so how to make a chromosome? Actually, it starts from the DNA. So you see all this DNA? This is your DNA polynucleotide chain, all right? This one. Now, it's not so simple. It has to be wound. Wound means you ikat, ikat around what? It is going to belit, belit is to wind it around 8 histone. So there are 8 proteins here, 8 molecules of protein here, and they call it a histone. Histone molecule, right? This one. Histone here. Wait. Here. Okay, sorry. Okay. So this one. This is a histone. 8 histones. This is a protein. Histone is not protein. And the DNA wound around the histone will form the nucleosome. So this one is a nucleosome. Okay, this is a nucleosome. Okay, nucleosome. And then after what happens, the nucleosome are going to form tight coiling. It's going to intertwine. It's coil around and become very tight to form chromatin. So your chromatin is going to be a basic unit to form your chromosome. So later on, it will condense, become thicker, shorter and thicker to form this structure that you see here that you call chromosome. Okay, so all this genetic information is found inside your chromosome, and the chromosome is inside the nucleus, in the cell. Every cell, every cell will have nucleus. Every cell will have chromosomes. Okay, all right. So this is the end of chapter 4 actually. Okay, to explain to you the different types of organic structures. So you started off with carbohydrate, then protein, then lipid. Started on water first, then carbohydrate, protein, lipid, and lastly you have nucleic acid. All right, okay, so that is the end of today's lesson. So I hope that you have learned all you need to learn about nucleic acid. All right, so share this share this video with other students who may need this lesson. Okay, I hope to see you another time. All right, bye bye everyone. I think there's some questions here. Okay, see you another time. Thank you for watching. Bye bye.